Hello everyone. This video is for those of you who may have been absent in class the day that we discussed how to approach the blood typing activity. Essentially what has happened is there has been a crime, there's been blood left around the scene, and we have to test that blood to determine really what happened at that crime. So you're kind of playing the detective here. Before we get into that particular lab activity, I wanted to touch base with you on the blood types. There's 4 types. There's A, B, and O, and each of these has a positive and negative form, so actually there's 8 different blood types. It is important to note moving forward, and this is something that I see get confused every year, type A positive blood and A negative blood, they are not the same blood type. The only thing they have in common is they both have the A antigen. So do not get that mixed up. A positive, A negative, they are 2 entirely different blood types. Same with B positive, B negative, so on and so forth. Recall that blood types are based on what antigens happen to be present on the red blood cells. Type A blood is called type A blood because it has the A antigen. Type B blood is called type B because it has the B antigen. AB is called AB because it is both the A and the B antigen, and type O is type O because there happens to be no antigen. Also recall our discussion on antibodies. Antibodies are present in blood. Antibodies will attach to antigens that are recognized as being foreign. So when we look at type A blood, we can see that the A antigen kind of has a lock and key mechanism, and this B antibody has a lock and key mechanism either. They will not fit together. However, if we mix A blood into a person with type B blood, the B antibody will attach to the B antigen. That would be a lock and key mechanism that would fit. Should that happen, the blood will clump up or agglutinate. So what we're going to be looking for in our lab activity is we need to use anti-serums to figure out what antigens happen to be present in blood. Familiarize yourself with how to determine a blood type. Let's go through a little practice problem here. In order to determine what blood type this is, we have to use what are known as anti-serums. An anti-serum is essentially a collection of antibodies. So if we have an anti-A serum, that actually contains A antibodies. And recall from what we saw on that last screen, A antibodies will attach to A antigens. So I can test this blood by taking a sample of it and putting it into this little cup over here. I can then take some A anti-serum and add about 10 drops to that. And I can start to see, is this agglutinating? If this is agglutinating, that tells me that the A antigen is present in this blood, and by default, this then could be type A blood or type AB blood, because both A and AB blood have the A antigen. It could not be type B blood or type O because neither of those have the A antigen. So if I start to swirl this a little bit, I can start to see that our blood is starting to thicken up, which is clumping or agglutinating. I now know that this possibly is A or AB blood. Now, we have to test to see if there is the B antibody, or B antigen. The anti-B serum will have B antibodies. So let's set this blood sample to the side, get the A serum out of the way, and here's some B serum. Let's check that one out. Same thing, squirt, and about 10 drops of our serum. Swirl a little bit. And it clearly is not solidifying like the other one. That tells me that this blood does not have the B antigen, therefore this automatically is type A blood. If this had clumped up here, I would know that it had both the A and B antigen, and I would know that is type AB blood. So now I determined that it's type AB blood, but is it type AB positive or AB negative? To do that, we would need a third serum known as the Anti-Rh Serum, which contains the Rh antibodies. Running the exact same test, I can determine, does the blood clump up when I add the Rh? If it does, that means that it is a positive blood type. If it does not clump, that means it is a negative blood type. So I could take another sample, take my Rh antiserum, add about 10 drops, and give it a swirl, and I immediately see that it's starting to kind of clump up. That tells me that this does have the Rh antibody or antigen, so I now know that this blood type is AB positive. I'm sorry, type A positive. So what you'll need to do to actually get this lab started is you'll need to complete an assignment showing me you understand how to do this before I actually give you the okay green light to run wild with this. This is the front side of your assignment. I'm telling you that these are the different blood types, type A positive, A negative, so on and so forth. What I need you to do is let me know what's going to happen with A positive blood if you mix in the anti-A serum with it. Will it clump or not? How about when you mix in the anti-B or the anti-Rh? So you will be putting essentially pluses in each box where agglutination would occur and minuses in the boxes where no agglutination would occur. Once you get this chart done, you can check out these bottom 5 questions. This one I'm telling you that blood will agglutinate with the anti-A and the anti-B serum, but nothing more. What blood type would this be? Same thing with 2 through 5. Each of these will have exactly one answer. Once again, when you get this page done, bring it up to me so I can quickly glance it over and steer you in the right direction if you happen to be confused with it. And then after that, you can turn the page over, read through the scenario, and then begin your investigation.